guess there are a few things that didn't make it up on the web yet. Soon they will be, thanks to this film in here. Okay, I encourage interaction. If anybody wants to stop and ask a question during my talk, feel free to do that. Okay? If it runs over time, is there going to be a bunch of angry med students storming in here at exactly 1 o'clock? Is that true? Does anyone know? Okay. The other frontiers, was their class after?
Just climb over.
Yeah.
Well, at 1.
So, you already heard about the laboratory for neuroengineering. This is 7 or 8 other professors who are working where technology and brain tissue come together. Me being the director of it just means that I have to do more work managing the place in really kind of a leadership role. You know, with that, it's like herding cats. So, however, this entity has been around as long as I've been in Georgia Tech, for about 10 years. It was founded by Steve Wirth and Bill Ditto. And as far as I know, it was the first neuroengineering center in the world. And now there are quite a few of them here and there. So that's my plug for the NeuroLab. It is mostly part of the lab, mostly part of the Coulter Department of Biomedical Engineering. There's a couple of people who are also electrical engineering. And that, as you may or may not know, is a department that's shared with Emory University. So not only do I have a faculty ID for both universities, but most importantly, I have a parking permit for both universities. So this is our model system. The experimental animal that we work with is a dissociated primary neuron culture that also contains glial cells that we get from rat embryos, usually late-term rat embryos, day 18 or so. This is a time-lapse movie that I made when I was in Jerry Pine's lab at Caltech that shows 4 hours in the life of one of these cultures just after it's plated. You can see when the movie starts, they're just little round balls. Whatever connections those cells may have had when they were in the embryo have been brutally ripped off in the process of dissociating the cells. And amazingly enough, within hours, very few hours, they start regrowing. They're following some genetic program to form connections with each other. And if you watch this movie carefully, which I've done for far too long, you will see that sometimes these growth cones reach out and then retract. So they're not just making connections with whatever neurons they happen to bump into. They're being somewhat selective and trying to form a functional network of some sort based on whatever genetic programming they happen to carry with them into this strange planar environment that they found themselves in. So not only do they form morphologically a network, but they also form an electrical network, electrochemical network, you could say, that has a lot of spontaneous activity. So this is what that sounds like if you record their activity using one of these multi-electrode arrays, and I'll show you in a moment. So every little tick you hear on there is one neuron firing one action potential recorded extracellularly by an electrode that the cell is growing on, or perhaps one of its axons is growing on. So this experimental animal that we're using, these multi-electrode arrays with cortical neurons growing in them, have a number of advantages. Compared to real brains, they're much, much simpler, 50,000 versus 100 billion neurons or so. So that means that there's pluses and minuses with that. Most of the important structures of the brain are not there in vitro. But we have much more control over all of the variables. We don't have to worry about hormones and circadian rhythms and menstrual cycles and all those things that real animals have that make it hard to do experimentation sometimes. We have much, much more control over those things. We could have just done this all in modeling in a computer simulation, but I truly believe that the complexity of real live, wet, squishy neurons with all their chemicals is important in how the brain works. And presently, we can't model that yet. It's too complicated. I'm not saying that all modeling is worthless, not in the presence of Dieter and Astrid. There's many useful things you can do with modeling, and in fact, we do in our lab some modeling ourselves. But I think, and they would probably even agree with me, that there are certain things that are still mysterious about living networks that are probably important to their function. So, and by taking the neurons from mammals, we can say that some of the things we see in vitro are related to mammalian nervous systems. Another really important, and probably the most important aspect of these cultures that makes them useful is their accessibility. To have a monolayer of neurons and glia growing flat on a glass surface makes them extremely accessible to imaging, to pharmacology, electrical and optical stimulation, as you'll see in a moment, genetic manipulation, physical manipulation. You can do all different things with these that would be difficult. I'm not saying impossible, but it would be difficult to do in vivo. So you'll see a few cases where we took advantage of those properties of these cultures. So the cultured dishes that we grow them in look a lot like this one here, with 60 electrodes embedded in the substrate. You can see along the, on the shadow on my hand here in the perimeter are the contacts that plug into a preamplifier that sends the data into a computer where it's digitized. Here's what it looks like when you zoom in on the network. We grow these networks quite densely, probably on the order of a couple thousand neurons per square centimeter, per square millimeter, excuse me, and they're pretty much filling all the open space on the dish there so that we can enhance the probability that any given electrode will be interacting with one or more neurons. This is a nice graphic that my former grad student, Andrew Chow, created that describes pretty much the entire lab. We have the multi-electrode array culture sitting here on a microscope that's got an enclosure built around it. We insulate our microscopes so that we can keep them warm, put them in a proper atmosphere with 5% carbon dioxide to keep the pH correct. We basically turn the microscope into a cell culture incubator. This microscope also has cameras on it. There's a very high-speed camera that we use for voltage-sensitive dye imaging. We created the first, as far as I know, the first closed-loop system using cultured neural networks, in which the activity that's recorded is processed by the computer in real time, very quickly, and used to decide what kind of electrical stimulation should be delivered. Let's see if I can do this with my cursor here. What kind of electrical stimulation should be delivered to the cultured network over here? So you could think of the recordings that we're making as motor commands, and they could be used to drive a simulated animal around on the computer screen. That's called a neurally controlled animat. Or they could be used to drive a robot around. And with my postdoc, Tom DeMarsh, we were the first ones to actually take cultured networks and use them to control robots and simulated animals. Okay, what can you do with these things? We have a number of big questions, which we may or may not ever answer, but we're chipping away at them at least. I suppose the most important big question, and especially when I'm wearing my neuroscientist hat, is what changes about brains when we learn something? Hopefully, when you walk out of here today, your brain structure will be a little bit different than it was when you came in, and it won't just be because you ate lunch. Hopefully, you will have learned something. A few synapses were formed, so maybe some of them were pruned they didn't really need anymore. Something, those lyrics from the 70s, I wish those would get pruned out of my brain and leave some more room for science. I know all of the songs from the 70s. How do neural networks process and store information? So this culture dish has a functioning network in it. Functioning is, up to interpretation. But we think we can use it to process information, and because it's so accessible, we can study that in more detail than one can in vivo. And can we take some of the ideas of what we learned, and now I'm talking more from my engineering perspective, can we take some of those ideas of what we learned and apply them to artificial intelligence, perhaps making computers a bit more biological, make artificial intelligence less artificial. We have a project that I'm not going to talk about at all today that's a collaboration with some power engineers to figure out how to control the smart grid of tomorrow in the face of varying power supplies like wind power and solar power. It's going to be much, much trickier, and we're going to need new control strategies, and they are hoping that we come up with ideas based on living neural networks to control those with. So, and then near the end of the talk, if I have time, I'm going to get into a little bit of philosophical stuff about how we should think about these semi-living creatures. We have robots, mechanical devices that are being controlled by living tissue. So we call it a semi-living creature, or hybrid, neurally controlled animat, is all the names we use. I conceived of this while I was a grad student at UC Irvine, and thought I wanted to study learning and memory in some system that's accessible, and perhaps if we have such a simple system, we can study it from both directions. You know, with a culture dish, you can do all sorts of molecular and biochemical and genetic studies, looking at it from the bottom up, and when it's controlling animat or a hybrid, it can behave. It can actually do things and learn and accomplish goals. And so you could study this from the top-down approach if you wish, and conceivably both. You know, you could actually, neurally controlled animats could bridge this gap. And because of the accessibility, we hope, and this has been my plan now for over a decade, to watch the learning process while it happens. I hope that the next time I give this talk, I will have more results to show you in that regard. Right now, I just have beautiful pictures to show you that our 2-photon microscope that we built with Georgia Tech does actually work, but the environmental control system hasn't yet been perfected for doing these long-term experiments where we want to watch the learning over the course of days or perhaps even weeks. Okay, so what I will talk about, instead of the big picture questions, I'll focus a little bit more on some specific questions that my students are working on. In this case, it's published work from my previous students, Doug Bach and Zanis Chow. Can we train cultured networks with multi-electrode stimulation? And I probably won't be telling you this unless the answer is yes. Can we control pathological bursting in vitro and in vivo? That is still to be determined. How can we optimize electrical and optogenetic stimulation? Those are two different projects, both oriented towards making better interfaces to neural networks. And what are the mechanisms of homeostatic plasticity, which is a collaboration with Pete Winner, who I saw here somewhere. Where is he? There he is. And it's thanks to the student Ming Fei Fong for bridging the connection between the two of us there. So first I'll talk a little bit about the learning in vitro stuff. Can we train cultured networks with multi-electrode stimulation? In order to make this possible, we first had to develop some kind of multi-electrode stimulators. There weren't available when we started this business in around the year 2000, any kind of stimulator that can stimulate on more than 8 channels. And my postdoc Tom DeMars built one that could stimulate on all 60 channels. Daniel Bakadar made another one. Actually, I think this is Daniel's one. This is another one of Tom's. And in fact, John Ralston, another graduate student, made yet another stimulator. So we have a number of these multi-electrode stimulators that can deliver arbitrarily complex patterns of stimulation to all of the electrodes. We feel like that was important in order to faithfully re-embody these cultures in some way and give them some artificial sensory system. To provide them with complex sensory input required us to make these special stimulators. So here's the results of the learning experiment, which I'll describe in a moment, but it's good to just show you that it did work first. This is a learning curve that you can see takes about more than an hour or so, to get up to whatever the top amount of learning is in this experiment. And this is an aggregate of a number of experiments all averaged together. And this red line here represents an important control experiment that we did in which we didn't have a closed loop. So the networks were undergoing some kind of learning protocol. They were getting stimulated just like these ones up here, but the stimulation was in no way related to what they were doing. It was just, you could say, think of it as a yoked control experiment. And then after the networks learned, this is a forgetting curve, we turn off the learning process and just let the thing behave and see how long does the behavior still maintain what it was supposed to do, and the answer is for about an hour and a half or so. So the kind of learning we were able to induce in these with multi-electrode stimulation is something that lasts on the order of an hour and a half. Typically is on the order of how long long-term potentiation lasts. So what was the task? We have these animats running around on computer screens. They're running around in a field here, and they were this describes 4 different experiments here in which the animat was supposed to run in the direction of the black arc there. So in this case, it was supposed to go in the southwesterly direction. And this bubble here represents what it did at the beginning of the experiment. If you were to zoom way in on the center here, that's what you would see. So it's kind of running around randomly, but after a while, it does learn and starts to run in the right direction. And then here's another experiment that was supposed to go through the upper right-hand corner, and eventually it did. And in this case, it started off going in the wrong direction, but eventually changed and learned to go in the right one. Here are the open loop controls, in which sometimes it may have accidentally learned to go in the right direction, but other times it's just wandering in the completely wrong direction. So that's the behavioral task. We set forth a goal for these networks. They had to move in a certain direction, and they had to move a simulated animal in a certain direction. It's a very simple task. There's only one thing they had to learn at a time, so our demands on the culture were not very high. And I'm sad to say that this is about as much as we could get them to learn even to this day. That was around 2007 when we did that. So this is the closed loop system that we use to do that learning. If you send an electrical pulse to the network, which we call a probe pulse, you see the word probe down here, this blue arrow represents 1 little millisecond long zap of electricity that causes the network to fire a bunch of action potentials. You pick those up on all these electrodes, and you make a vector that points in a certain direction based on where the activity is in the dish. Okay? We call that a center of activity, just like a center of gravity. If you have a plate with various things on it, it's got a center of gravity somewhere. Center of activity points in a certain direction. We could use that to move the animat in that direction, but what we wanted to do was to start the experiment by subtracting away any bias that the animat may have at the beginning. So it may have a tendency to wander in the upper right direction at the beginning of the experiment. So we create a matrix that normalizes its behavior and makes it equally likely that it will run in any direction. And this matrix is frozen in whatever value it was at the beginning of the experiment. So then you run, when you ping the network and it gives a response, you run it through this matrix, and then you get a vector that tells the animat which way to move. And then you ask the question, did it move in the direction we wanted it to, or didn't it? And if it moved in the right direction, then you deliver this shuffled background stimulation, basically just sprinkling a little bit of stimulation across the network, and that seems to preserve the synaptic weights in whatever values they were in. When it moved in the wrong direction, you deliver what's called a patterned training stimulation. So this is pulses of electricity that are sequenced with very specific timings to take advantage of spike timing dependent plasticity. And if you repeat those sequences a few times every cycle, you can shape the behavior of this cultured network to control an animat to move in a certain direction. This shows a little bit more detail about that shaping process. So you could think of those PTSs, the pattern training stimulation, as little melodies that we play to the network. And Doug Bauckham and Zenas Chow had this brilliant idea to create a library of arbitrary melodies and start using them to train the networks at random, and the ones that work we use more often. So they bubble up to the top of the list, and the ones that don't work in training, they go down to the bottom of the list. So this graph here just tells you what the, the rule is for moving them up and down, the probability of being used. So, and this whole cycle lasts about 6 seconds or so. During the training part, which is 4.2 seconds, you're repeating one melody over and over, several times, and reinforcing activity on specific circuits in the network. Probably strengthening some synapses and probably weakening others. The main thing is that you're just changing the functional connectivity of the network in some way or other. It's interesting to note that during the learning process, any given PTS that worked may not work later on in the learning process. In other words, we keep having to reshuffle what order we use the PTSs. Some of them are good for training the network at the beginning of the experiments, and then they become less good. Ones that weren't any good at the beginning will change and become more useful. So the take-home message there is that this is not a stationary network. It is constantly changing. It's alive. It's dynamic. It's rewiring itself all the time, as you saw in the time lapse at the beginning. And therefore, any training strategy that we want to apply, if we're going to use this for other things, has to be adaptive. It has to be not only closed loop in the sense that I've described here, but it also has to be continually changing to take into account the way the connectivity keeps changing in the network. So conceivably, there could be applications for this for recovery from stroke. You can imagine multi-electrode arrays implanted in stroke patients that could selectively reinforce certain circuits that are remaining in the person's brain to take over functions that were lost when the tissue got damaged by the stroke. Yes?
Did you ever look at, developmental issues as far as this is concerned? I mean, like when you're saying it changes, I think you're talking about over hours or a day in a fairly stable network, or do you apply it at different points in the days in vitro?
I know that this type of learning experiment only worked when they waited for the culture to be at least 3 weeks old or so.
Really?
Yeah, so they had, I think the reason for that was the culture had to have differentiated firing. And we have a paper in which we sort of created a taxonomy of the firing patterns of these cultures. And during the first 2 weeks, they go through a very stereotyped periodic bursting kind of phase, which is something that you see in the spinal cord of chimps as well. And the network is behaving as a unit. When they get older, there's differentiated patterns of activity, so you could imagine that that can represent more information if the thing is spatially differentiated. That said, we did not do a proper study of this, learning as a function of age. I think it needed to be done. It will get done at some point.
If you catch the system when the forgetting is not complete, could you take it in a certain stage, change the T, and then reinforce the memory to have a longer duration?
You probably could. Certainly stopping training and then restarting it before it forgets, you get some advantage, because the behavior is already moving in the correct direction there. So the retraining would be easier than it would be from a completely naive network. But how much advantage and what are the limits of that, and whether it would make it remember longer, I don't know. I kind of doubt it. As I mentioned, when the thing is moving in the right direction, we tended to deliver this random stimulation. One of the reasons we did that was because it was a control that has the same number of stimuli as the pattern stimulation. We wanted to make sure that it really was the pattern that was doing the training. That random stimulation does not bias the network to learn in any particular way, but it probably is making the memories fade away. So as it's getting a random background stimulation, it's probably unlearning what it was trained to do. Okay, so let's talk a little bit about the bursting as pathology. You can imagine that these population bursts that I showed you are something that result from deafferentation. In other words, you're cutting off the sensory input of the network, and it's no longer getting the input it wants. The network becomes hyperexcitable thanks to homeostatic mechanisms that all neural networks have built into them. If they're not getting enough input, they turn up the gain on the synapses or on some part of the system to increase their excitability. If you were to put an electrode into an epileptic person or a rat during a seizure, you would record this kind of simultaneous population bursting. Synchronized activity is a hallmark of epileptiform activity, and it's also inconsistent with conscious information processing. In other words, when you have this kind of activity pattern, you're not processing much information. Either you're having a seizure or you're in slow wave sleep. That said, that kind of activity pattern during slow wave sleep might be something that helps reinforce memories, but you're not having a conscious thought process like you would during REM sleep. So, my first graduate student, Daniel Walkenaar, came up with a scheme for quieting these bursts by restoring some kind of the sensory input artificially with these electrical stimulation systems that he and other folks built in our lab. So here you see what happens if you stimulate on a single electrode. It's 10 stimuli per second, you get rid of the bursts. Occasionally one of them happens, but you get rid of them for the most part. If you stimulate only one stimulus every 10 seconds, you can entrain the burst. You can get the network to burst every time you stimulate it. So if you wanted to do that for some reason, you could do that. If you stimulate on many electrodes at a total of 20 stimulations per second, in other words, just 1 to 2 hertz per electrode across 10 electrodes, you can completely get rid of the bursting. You'll notice that the background firing rate is elevated, though, compared to what it was over here. So it's not that the network has been silenced. Far from it. It's still firing a lot of action potentials, as you'll see in the next slide, but it's not firing these synchronous population bursts. And then you turn off the stimulation and it goes back to firing those synchronous bursts again. So we found that when we used a single electrode to do this, we could only get it down just so much. We could not get rid of the bursting completely. Apparently that one electrode, if you're hammering it at 20 hertz, the cells that are near it tend to get tired out. They run out of neurotransmitter or docked vesicles or whatever it is. They're no longer as effective. Whereas if you're using multiple electrodes and you're only stimulating each one of those only at 1 to 2 hertz, they don't get tired out and they continue to be effective at quieting the network. So here's a movie that John Newman made of one of these quieting exercises. Here's the population burst here. Here comes another one. See, I'll turn on the sound. That was a population burst. And there he turned on the multi-electrode stimulation. And it's keeping the firing at an even keel here. You can hear it kind of sounds like melody, because in this case, it's a periodic, it's cycling through a number of electrodes periodically. So Daniel Hawkenar found you could just do this periodically at a set voltage, and it's effective. It works to quiet the networks down. But what he found was it works even better if you continually tweak the voltage according to how much firing the network has. So he's got a feedback algorithm here that's looking at the total amount of firing the network has and adjusting the voltage. If it's not firing enough, he turns up the voltage a little bit. If it's firing too much, he turns it down a little bit. In that case, you can have complete burst quieting for as long as you want. It'll just keep on not bursting. And you can also see that there is still this grayness here means that it is firing action potentials, just not synchronized ones. These black blotches here are each population bursts. Okay, so when we published that around 2005, everybody said, wow, maybe this would work to help people with epilepsy. And my then graduate student, John Ralston, found Bob Gross here at Emory, who is a surgeon who actually works on people with intractable epilepsy, and he also does research in his lab on that. And we started this collaboration to try it out in rats first, and if it works in the rats, then he may be trying it out in his patients. So you can imagine if you're faced with undergoing surgery where they're going to remove a fairly large piece of your brain to get rid of your intractable epilepsy, you may rather have some electrodes implanted instead, because if they don't work, you can just remove them. You're not missing that big piece of tissue like you would when you undergo the kind of surgeries that Dr. Gross does. So that's why we think that it's a promising idea that a drug-free treatment for epilepsy is something that probably would be welcomed by a lot of people, even people with epilepsy the drugs work for. They have a lot of side effects. So Sharon Desai is another graduate student in the lab that's co-advised by Dr. Gross, and she is one of the people that's trying this scheme out in rats. They're using an epilepsy model in which they inject tetanus toxin into the hippocampus. The hippocampus is part of the brain that has to do with storing declarative memories or events that happen in your life. And when people get epilepsy, it's often the place where the focus of the seizure is. It's part of the brain that tends to be the source of epilepsy. So it's a good place to have a model for epilepsy. We know where the seizure focus is because it's the place where we injected this toxin. And when I say we, I mean Sharon, who's an excellent neurosurgeon at this point on rats. She's testing out the difference between sending in this kind of distributed stimulation on multiple microelectrodes that are implanted, permanently implanted in the rats, versus a single macro electrode that's on the order of 0.5 millimeter across. These ones are about 30 microns across. First of all, if we look at recordings from these electrodes in two different animals, a control animal versus an epileptic animal, when they're exploring things and whisking and doing the things that rats do, they often have this oscillation in the field potential that's called theta oscillation that tends to be around 7 to 10 hertz or so in rats. And in the epileptic rats, there's not very much of this theta. So here's a power spectrum that shows the epileptic rats don't have this peak here at the theta that the control rats have. So she hypothesized maybe if we just stimulate them at theta frequency, about 8 hertz, we can get them to recover in some way from their seizures. One of the first things she did was to say, what is the range of influence of one stimulation electrode? And she did this using CFOS immunohistochemistry, which is basically, it was one of these immediate early genes that's expressed when you stimulate neural tissue. So she zaps some tissue in one of these animals and then fixes their brains and slices them and stains them to see whether CFOS was expressed and how far away from the electrodes it was expressed. And what you see, not too surprisingly, the macro electrodes have a bigger radius of CFOS expression than the micro electrodes. So you can get more localized activity patterns from stimulating the micro electrodes than the macro electrodes.
Steve,
Yes?
is the motivation for comparing to the macro electrode what the setup that's used in DBS? Is that
Exactly, yeah. So this macro electrode isn't quite as big as a human DBS electrode, but it's big, you know, compared to the micro electrodes. It's got a much lower impedance, and the kind of electrodes that they implant into people, like Bob Gross does this for people with Parkinson's disease, are about 1 millimeter in diameter, and there are 4 electrodes per probe that they implant in your brain, and they usually implant one on each side. So those things have a sphere of influence that's going to be on the order of probably 1 millimeter or so, whereas the micro electrodes, it would be a much smaller sphere of influence. Yeah.
Does the single big electrode give a bigger charge than each individual micro electrode?
It does, yes. Because it's a low impedance, you can pass a lot more current through it without causing damage in electrochemistry to happen. So you are delivering much more charge from one of these macro electrodes than you would from a micro electrode.
But is the idea that the sum of all of the charges delivered by the micro electrode array is equal to the big one?
That is exactly the idea. Not only would the sum of all the charges from the micro electrode be equal to the macro electrode, but it's more spatially defined. You could conceivably make the micro electrode shaped in such a way to maximize its effectiveness. And if you put in enough electrodes, you could use only the ones that work. You know, you could select, you could say, okay, let's just pick the top 20 electrodes out of 100 electrodes here that do the best job in helping whatever the problem is, whether it's epilepsy, Parkinson's, or whatever. So Sharna has tried out a variety of different kinds of stimulation. This one, she's synchronously stimulating all 16 of the electrodes, boom, 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 like that, at 7 hertz or 8 hertz. Here she's sending in a sine wave through those electrodes, and here she's sending in random pulses, more like that quieting stimulus that you heard a minute ago. And these graphs here show what happens when you watch these rats while they're undergoing the stimulation. That's the point in the middle. This is their pre-stim level of seizures, and this is their post-stim level of seizures, and this is the stimulation seizure level. What we saw, at least for these guys here, was that there was a statistically significant reduction in the number of seizures that they had while the stimulation was turned on. Same thing went for the macro electrodes. There was some improvement with this sine wave stimulation that was statistically significant. For the micro electrodes, it worked whether it was intermittent or continuous. For the macro electrodes, it worked better if it was continuous. And this synchronized didn't work very well at all. So there's still a lot of parameters to be tested. She did not cure any of the rats of their epilepsy. They still did have some seizures. One of the nice things about the tetanus toxin model is that these rats, number one, it very reliably causes them to form epilepsy after a few days. And they have seizures about every half an hour or so. So during an experiment, it's pretty sure that they will have a seizure or two. And then they recover. They actually get better after a month or so. They no longer have epilepsy. Unlike some other epilepsy models from rats in which the animals get worse and worse and eventually die from it. Okay, so I'm going to move on to the electrical, how to optimize stimulation. And first I'll talk about electrical stimulation, and then I'll talk about optogenetic stimulation. So Michelle Kuykendall in the lab has been doing optical recording of fluorescent signals that we get from calcium-sensitive dyes. So if you pour on a certain dye on these networks, the fluorescence is proportional to how much calcium is inside of the cells. And when cells fire action potentials, they have calcium channels open up and let in calcium, and you can see them flashing here. So those flashes represent probably bursts of action potentials. The response time of calcium dyes is tens of milliseconds, so it's very difficult to see individual action potentials with this kind of imaging, but you can definitely see bursts of action potentials, and most of those flashes, what you see there. In fact, when you see the whole thing flash, that's one of those population bursts that I showed you before. So this is spontaneous activity, and what Michelle is studying is electrically evoked activity. So she has made a very complicated technological system here using MATLAB and all of the hardware that we have in the lab to look at the movie that she records from a high-speed camera of the fluorescent flashes, to look at those flashes and decide how effective was the electrode at stimulating various neurons in the field of view. And if you want to stimulate this neuron over here and it didn't fire, this algorithm here decides how to change the stimulus to increase it or make it longer in order to get that neuron to fire. So she has this closed loop system, which is basically doing the job that a graduate student normally does. Normally a graduate student looks at the movie, they process the data, and they go, oh, I think I need to turn up the voltage a little bit. And they do that, and then they try the experiment again. And that process takes days, many experiments. She can do all this in one afternoon with one dish by just pushing a button and saying go. The thing automatically optimizes the stimuli to get it to work. And here's an example of that. So here's some neurons sitting in a dish next to two electrodes. Let's imagine that these are the two neurons that are depicted in this graph here. They're near each other, and you want to say, how would I best stimulate the blue neuron with electrode 1 versus with electrode 2? So here's electrode 1. The automated algorithm is going through various different stimulation currents, and it's doing them in random order to see what's the firing probability. And you can see in this case, the blue neuron fires much better with stimulation from electrode 1 than the dark blue fires better than the light blue neuron. Electrode 2 is better at firing the light blue neuron than the dark blue neuron. So she can get stimuli. She can figure out what stimuli will effectively stimulate this neuron versus that neuron, and what value of stimulus and what waveform should she use to get the optimal stimulation. You can imagine that sometime in the future, when she gets really good at this, she'll be able to just look at the electrical recordings to figure out what the responses are and not have to do the optical imaging. That will be great, because then we could do this in an animal or a person's head. You could imagine optimizing the stimuli this way by recording the responses and carefully adjusting the stimulation on different electrodes to be able to send the stimulation to different circuits in the network. That's where we're going with that. That's a collaboration with Steve Dworth and also Martha Grover at Georgia Tech. So now I'm going to talk a little bit about optimizing the optogenetic stimulation. So how many of you here are familiar with optogenetics? Okay, I hesitate to ask this. Is there anyone here who has never heard of optogenetics? I know it's embarrassing to raise your hand in those circumstances. It's okay. Thanks mostly to Carl Neisseroth and Ed Boyden. This is now the hottest topic at the Neuroscience Conference that a lot of us just came back from. What the idea was to take genes that they got from pond scum, as Michael Korath says, algae and bacteria, take genes that made those organisms light sensitive and splice them into neurons. Seems like a strange idea, but the cool thing about this is now the neuron becomes light sensitive. Never was before. These are not normally light sensitive cells, except for if you put too much light on them, you could kill them, obviously. But in terms of turning them off and on with light, these genes work for that really, really well, thanks to a lot of hard work by these guys who mutated them to make sure they would work well. Different colors of light activate different genes. So this one called channelrhodopsin is activated by blue light, and we can use it to make neurons fire. This one called halorhodopsin is activated by yellow light, and we can use it to stop the cells from firing, to inhibit them. And I won't go into any more details about that. You can read about it. There's lots of stuff out there. Something else I should mention, though, is that you can put promoters in front of the genes to make them expressed only in certain cell types. So in this case, the CAM kinase 2 promoter allows it to be expressed only in excitatory pyramidal neurons in our culture. So that might be handy later on. We're planning to have a variety of different promoters and have these things expressed in a variety of different cell types. We haven't quite gotten there yet, but even just with the CAM kinase 2 promoter, we've done some amazing things. And when I say we, I mean John Newman here for the most part. He built this optical stimulation rig, which has very powerful LEDs connected to fiber optics, which are then connected to this set of optics here that scrambles the light to make sure it's a very homogeneous illumination on the culture dish here. That's called Coulter illumination, which is something familiar to most microscopists. And he also built an electric circuit to make sure that when you command the LEDs to be a certain brightness, you do get the brightness you commanded them to do. So here's the commanded brightness, voltage, and here's the actual light that you get out of it. It's a nice linear response thanks to the fact that he built a very good feedback circuit that uses a light sensor here. So it's constantly looking at the light output and adjusting the value to make sure that it's what you think it is. Here's, for those who are extreme geeks in the audience, here's the circuit that he built. It's got this feedback photodiode here, and this is just to show this, and this curve here to show that it's a very fast circuit. You can respond. When you give it a command to turn the light on, it turns the light on very, very fast. This is a 5 microsecond time scale here. So it's on the order of 1 microsecond, which is far faster than any neuron ever cares about. We're usually in the millisecond regime in the neural world. So because it's so fast, you can provide the neuron with light stimuli that are quite complicated. You can make them as arbitrarily complicated as you want, like these waveforms right here that have this sort of Gaussian distribution. This is a Ornstein-Uhlenbeck process with a 50 millisecond time constant. And that will become important in a moment. So in collaboration with Tatiana Timochenko, who is in Larry Abbott's lab, soon to be in her own lab at Max Planck, John has been, and also with the help of Ming-Fei Fong, has been stimulating networks of optogenetically transfected neurons with these complicated light signals, these OU signals. And what you can do is you can play the same random noise signal to the network over and over again. You know, it only lasts a few seconds. You play it over and over again, and you look at the response across trials, and that's what's plotted vertically on each one of these little plots, raster plots here, under different pharmacological situations. Here's an active network. Here's one in which he's blocked the glutamatergic neurotransmission. Here's one in which he blocked that and also the GABAergic neurotransmission. And you can see that it's much more reproducible activity when you start blocking these, when you're basically creating a network where each neuron is an island unto itself rather than talking to all of its neighbors there. So there's a huge number of interesting things you could do with a system like this where you can apply these arbitrary stimuli. And this is just spatially homogeneous illumination. At some point we'd like to have spatially localized illumination to target specific parts of the network or specific cells. So another thing that we're doing with this that's I think, really powerful is to make an analogy to Daniel Bachenar's burst quieting, only it's an optical version. So Daniel, as I mentioned before, invented this closed-loop system in which the voltage is adjusted according to how much the neuron is firing. John made an optical version of that we're calling the optoclamp nowadays, in which the brightness of the LEDs is adjusted, or it might be the time, might be the duty cycle, or the frequency of the pulses. Some property of the LED's illumination is adjusted according to how much the cells are firing. And here he's trying to attain various different set points. And you can see here, this is the target firing rate, and this is what it actually fired. It really is very well locked. There's actually error bars on those plot symbols. I mean, it's perfectly locked to the value that he tried to set it to, except when you try to crank it up to too high of a firing rate. You're shining a lot of light on it in that case, and you can't clamp it anymore. At some point, the system obviously is incapable of getting to fire that fast. So this is a short-term optical clamping on the order of a minute or so at a variety of different firing rates. John has tested this out with much longer experiments. In this case, each one of these was a day-long experiment with one cultured network that he tried to clamp it at a variety of different firing rates. So 0.75 hertz per unit up to 6 hertz per unit. You know, the background firing rate is probably more like these firing rates down here. And the good news is that you can keep this up for pretty much as long as you want, and you could even do this for many days in a row without destroying the cells, without ruining the optogenetic construct. It keeps on working beautifully. So you can clamp it at a set firing rate. In some cases, again, if you try to clamp it at a too high a firing rate, the system is no longer able to keep maintaining that level. For whatever reason, either the cells are getting too much light and they're becoming unhealthy, or perhaps the optogenetic constructs are getting bleached out, or something like that. We're not sure what the reasons are, but as long as you keep it within some range, you can clamp the firing rate at a very specific level. Okay, and yeah, I wanted to say that that was a day-long experiment for each one of those. Yes,
What does that expression look like? Do you know whether it's expressed in all of the cells that you're targeting it to, and how uniform?
Yes, so when we transfect these, we use an amount of virus. The gene is carried into the cells by virus, sort of a disabled adeno-associated virus that is in big enough numbers that all of the cells are multiply infected. And only the ones that have the right machinery to activate the promoter that we put in there express the construct. So only the ones that would normally express CAM kinase 2